So without further ado, I see Diane has managed to share her screen. I hand over for the final presentation for our session to Diane Mostert. Welcome.
Hi Irene, thank you so much for giving me the opportunity to present today. It's been really interesting to listen to all the talks. So I'll get into it straight, as I see like we've gone a little bit over time already. So yeah, as you said, this is like one of the aspects that Altus's group has really focused on in the last 15 years or so. So there will be quite a bit of older research in here and some new things that we are doing. So I'll start with this slide just by saying that although half of the bananas in the world planted today, the Cavendish type, the banana is actually quite diverse, as all this audience probably knows very well. And as one of the most accepted theories of evolution of FOC relates to the host and the pathogen actually co-evolving in the center of origin, this is an important point of the part of my presentation today. As this co-evolution between the pathogen and the host really helped to shape its population structure. So it is therefore quite interesting to look at the evolutionary history of the host as well. And I think most people also know this, that the center of origin for banana is also the Southeast Asian region. And it is basically the result of hybridization between two subspecies. And as banana was one of the earliest crop that was domesticated by people, they moved it around all over the world as they moved to new areas. Particularly interesting for Africa is that some were moved to East Africa, where local adaptation gave rise to the East African highland bananas, which are kind of like very special to this area. It is thought that banana was also introduced into the Central American region much later from the Canary Islands by Portuguese colonialists, only in the 1500s. And of course, then the fruit became incredibly important, and they started a lot of large-scale banana production in Central America to export to the US in the late 1800s. And this of course, because the banana, this banana that it was based on just one variety at the time, Gros Michel, and because of the clonal means of reproduction of this variety, this made the variety very prone to diseases and pests. And as the banana industry expanded in the late 1800s, people started to notice mass decline of banana plantations. The first banana wilt epidemics in Latin America devastated production, with more than 30,000 acres abandoned in Panama and Costa Rica and so forth. And new fertile land in Central and South America was often opened up to continue production, only to be abandoned after a few years. And it's estimated that that by like 1958 or something like that, over 100,000 acres of banana had been abandoned in Central and South America. So it was really, really devastating. And it's therefore not a surprise that FOC was regarded one of the most important plant pathogens of the 20th century. Of course, ultimately in the 1960s, the whole industry converted from Gros Michel to Cavendish that we have today, and this solved the problem. However, as early as the 1967, it was also noticed on Cavendish, and it was, but only later in 1990, it really developed in a full-flung epidemic in the Southeast Asian region. Basically, it was mostly confined for around 20 years in the Southeast Asian region, but then around 2010 to 2015, it started moving to the Middle East. And also this is when it was introduced into Mozambique, where a lot of our research is focused in Africa. But since it's also moved to Laos, Myanmar, or all around in Southeast Asia, and there's more unofficial reports from other countries in Southeast Asia. But of course, one of the most devastating has been the recent introduction into Colombia in South America. And what I really want to say with this first part of the presentation is look at the large-scale production of Cavendish and really how this has like influenced the spread of this disease as the production area have been increased to different countries. And the same is really true for the Gros Michel. As Gros Michel, the industry grew in the early 1900s, so the pathogen spread. And this is basically the first point that I want to make, that the type of variety that's planted really influences the diversity of FOC populations that you would find in a certain area. Well, then I'll shift over to, I think, something that everyone knows, that FOC is basically part of the Fusarium oxysporum species complex, and it's a group of morphologically related fungal strain that is very destructive on different hosts. And the classification of this pathogen is really complex. Over history, it's been complex, and until today, there's more also new shufflings that's happening in the taxonomy of Fusarium that is hotly debated. I would leave it at that. Do not put my foot in it. But basically, in the past, that oxysporum has really been separated into these forma specialis, which relates to a specific host that it affects. So the one that causes the disease to bananas, of course, the Fusarium oxysporum forma specialis cubense, while others, like the one on tomatoes, called Lycoppersickia. And I think most people in this audience would know this, but more than 150 forma specialis have been described in the Fusarium oxysporum species complex. Then if we go into FOC, there's different races that is described. The race is defined by the ability of a particular strain of the pathogen to affect a specific group of cultivars or one particular cultivar. The race 1, of course, caused that early epidemics in Central America on Gros Michel, while race 2 caused disease to cooking bananas like Blago, and then race 4, which is responsible for the more recent epidemics on Cavendish, can also, unfortunately, also affect cultivars in other races. So then this is where it becomes quite confusing. Additionally, also the race, obviously, if you want to characterize a race, you have to do it with pathogenicity testing, which is very time consuming and expensive to execute. You have to have access to all the different plant or the host varietals, and it's often in conflict with what scientists find when they do field screening. And this is why vegetative compatibility really have been introduced. And this has been used quite effectively to measure the diversity of FOC populations. And I think Liz has also talked about vegetative compatibility a little bit, but basically vegetative compatibility is the ability of two different strains to come together and exchange genetic material. And for this to occur, the loci that governs this in the genome must be identical. So we assume that basically two isolates which are vegetative compatible is clones of each other. And this technique has been manipulated in the lab, and it uses a toxic media, a chlorate, to induce these mutants, and then we can kind of manipulate it to determine the diversity of FOC populations. 24 different VCGs have been described formally for FOC, and this is particularly diverse if we compare it to other Forma speciales in the oxysporum species complex. So then I'll go over to some research that we have done. This is published already quite a while ago, but we did like quite a lot of large-scale collections in Asia, this is just to iterate that the VCG diversity is really the greatest in Asia, supporting my earlier point, which suggests that FOC originated from the same continent where all the wild species of banana is also from. And the distribution of FOC VCGs in Asia is therefore also linked to the cultivars that are planted. And this is illustrated in this, this slide here, where, for example, in Taiwan, mostly Cavendish is planted, and you only see VCG 1316 dominating there. Whereas in Indonesia, where quite a lot of different cultivars are planted, commonly the diversity is quite much greater also in this regard. Of course, this is it's a bit of time ago. So now in other countries in Asia, we also identified TR4, and as like the industry is going over to different banana varietals, so does the population of FOC that dominate really change. So since this publication was published, TR4 is now also found in Laos, Myanmar, and Vietnam. And as I've said, it's really linked to that like expansion of Cavendish production area. So if we move over to Africa, which is where we do a lot of our research, of course, we have mostly a distribution of a group of very closely related VCGs. We call this the Fusarium oxysporum lineage 6. And these are found, or very commonly found, all over the East and Central African region. It's really, really dominated by these VCGs. And these are all kind of linked to race 1 and 2. And it's mostly found on susceptible cultivars, including Pisang Awak, Sukari and Daisy, Gros Michel, which are planted for the production of fresh fruits, as well as for beer, to make beer. But a lot of the varieties that are planted in this area, the East African highland bananas that I've mentioned before, is actually not susceptible to these VCGs. But unfortunately, of course, as again, Cavendish production was introduced, commercial Cavendish production was introduced into Africa in northern Mozambique. We actually found the first report in Africa of FOC TR4 here on 2 different commercial farms in northern Mozambique. And it's really been confined here in this area, haven't moved, luckily, to other areas, because just over the border here, Tanzania is very vulnerable to the introduction of FOC TR4. But most recently, we just published this paper with some colleagues in France. They also found FOC TR4 in Mayotte Island. This is a French overseas department. And as you can see, there isn't really that much distance between northern Mozambique and Mayotte. So we can kind of hypothesize that it could have been introduced, this TR4, from Mozambique, but this of course needs to be confirmed. And there's obviously also other possibilities of introduction. Very interestingly though, is that here we actually found it from, not from Cavendish, but we found it from a different variety, which was a very, very interesting finding. Then if one more interesting thing, this is a paper that I'm working on currently to publish. It's almost there to be sent in for publication. Just to mention that, like, if we go back to VCGs, once a point mutation occurs in those loci that govern vegetative compatibility, it could result in a new VCG. And here it's just a summary of this work that I've done, but this is it shows the presence of as much as 18 additional VCGs to be present. And this is a combination of work done by Susie Perry, which is an Australian researcher working for, I think, the Queensland Department of Agriculture. And she's going to be part of this publication, but we really found, we looked at her kind of new VCGs, novel ones that were not compatible to these 24 known VCGs, and we did some crosses with a new population that we investigated, and then we found that, yeah, there could be actually 18 more VCGs and a lot of single member VCGs that is present for FOC that is able to cause disease on banana. And some of these, for example, this one is actually quite important because we found it in 3 different countries, and it's particularly important in Oman. And further studies really need to be done on this population to see what is the virulence of these new VCGs, how widespread are they, and could they really pose a risk if we should keep our eye on it, basically. Okay, and then the last part of the presentation, I'll just shift to like more the genetic distance between different isolates and FOC populations. So this is also quite an old study by one of Altus's previous students. This study just showed that when we do phylogenetics with the core genes, the FOC Fusarium oxysporum species complex is divided into 2 phylogenetic species and 4 different clades, and that strains that cause disease to banana really fit into at least 2 of these clades, which would suggest independent origins, as Liz has also mentioned before. And here you can see that the ones causing disease to Cavendish are basically separated from the ones that cause disease to other varieties. And you can also see that these 2 are quite far removed from each other, with strains from other Formis specialis clustering in between the 2 clades. Then if we focus only on FOC, we will find that, as you see, here's the 2 clades in which FOC strains are separated. And each of these, basically, depending on what study you follow, we can definitely see that FOC kind of consists of 5 to 9 phylogenetic lineages, and within each of these lineages, closely related groups of VCGs are separated. Then I'm just going to go again to this work that we are almost publishing. Here we investigated a large amount of samples that represent all the known VCGs, as well as those newly described VCGs that I've mentioned before. And here it shows that there is high statistical confidence for the separation of 8 different morphic clusters of this FOC data set. And this was based on a next-gen sequencing data. And if we look at within each of these clusters, we did the PHI test that tests for the possibility of recombination to occur. And here you can see that most of these clusters were really clonal, with only one cluster showing some evidence for past recombination. But this, of course, could also have been due to parasexuality that is hypothesized to happen in FOC. If we go more into these clusters, here you could see that really the contrast between the clade A, this one including all of the STR4 and TR4 strains, while this cluster really includes the race 1 and 2 samples. And you can see quite a big difference, the separation between this cluster, which have all of the VCGs 4, 5, 8, 12, and 20. And a lot of these new VCGs are clustered here. And there's really quite a big difference between this cluster and the other clusters, which are, this whole cluster is really known to be the race 1 and 2 pathogens. Another interesting thing that we found is that there's one of these isolates that are actually phylogenetically distant from this traditionally clade A and B isolates. And this warrants a lot of more research, but it is quite an interesting finding. And then the last thing to mention here is this large diversity of VCGs that we really find in this particular cluster, that FOC lineage 6 that I've mentioned before. That's very common. It's quite widespread all over the world, but it's also the most important, apart from the new introduction of TR4, for example, of course, this one is also very important in Africa. Okay. So this, just to mention the last 2 slides, many studies have shown that the genes that we use, the core genes that we conventionally use for inferring genetic relationships of FOC, is poorly correlated with host-specific pathogenicity. And this basically comparative genomic studies have revealed that this is because the genome of Fusarium oxysporum is really compartmentalized, and it consists of this highly conserved core genome, and then which are conserved within oxysporum species complex members. And this is usually driven by vertical gene transfer events, while the lineage-specific genomic regions really encode the host-specific virulence factors, which could be unique for a particular forma specialis. And this has been linked to horizontally acquired, or due to various horizontal gene transfer events. And this is something that Liz has also mentioned, so, and I'll just quickly go through these because she's talked about this. And that these, so if you want to really look into where did pathogenicity towards banana really develop, you really need to look into those regions that are related to pathogenicity because of this incongruence between your core genes and because of this compartmentalization of the genome. And so far, really the only factors that have been studied well by Liz's group is these 6 genes. And she's really explained it like quite extensively, but it is important to note that we really need to look into novel effectors or pathogenicity factors that also have been mentioned by some of the other speakers, to really kind of tease out how pathogenicity towards banana have evolved. So to conclude, the FOC distribution can really be attributed to the host cultivar planted. And this is really evident from the history of epidemics driven by monoculture production, both on Gros Michel with the race 1 epidemic and Cavendish with the TR4 epidemic. And FOC is a particularly diverse fungus. We know there's 24 described VCGs. The clones varies in virulence. VCGs are useful to indicate the global spread, and there is even more genotypes, novel genotypes that are present, and we really need to look into these and what threat they pose to the industries. And then FOC strains have evolved onto at least two separate occasions, which we kind of can like separate from into the Cavendish and non-Cavendish phylogenetic that causes disease to Cavendish and non-Cavendish phylogenetically. And this has been influenced, of course, due with vertical and horizontal gene transfer events. And then lastly, to consider that genes that relate to pathogenic function, really we need to look into these to inform us on the evolution of pathogenicity towards banana. And that is it from me. I just want to say thank you to all our funding bodies and my group at Stellenbosch University, and of course to Professor Altus Viljoen, which is the head of our group. Thank you so much for the opportunity to present today.
Thank you very much, Diane. Yeah, and again, thanks to all of the speakers. The session has gone so quickly for me. I don't know about for everyone else, but sitting down at 6 o'clock at night for a session on biodiversity of banana pathogens would tend to indicate a long evening, but it's actually been remarkably short for me. Now, there are a couple of questions in the chat to Diane, or three. So from Elizabeth Aitken, Diane, do we know if these new VCGs are pathogenic, or could some of them be endophytes, is the question.
We did do pathogenicity testing, Liz, on those ones. So we know they're pathogenic, but what I mean is that like really that's just by small plant screening. So I really want to look at where they're present. Are they really a problem? Are they just like kind of some strain that's not really that genetically fit in the population? That's really what I mean by looking at like how important they are, but we know they are pathogens at least.
Okay, thank you. And from Tuan Nguyen, Diane, just wondering about your phylogenetic trees. What were built based on sequencing of what genes?
So the ones that I've shown in the presentation, the core genes, those were from Ghada Foury's study, and those were IGS, EF, and mitochondrial subunit. But of course, now we know that the RBP1 and 2 regions are really the important regions together with EF to do phylogenies based on the core genes. The other trees that are shown as obvious, the ones which showed the different separations, those were based on DART sequencing data. So
Okay, sorry.
data, yeah.
Thanks, Diane. And the final question to you from Tony Patterson. Diane, how does the production system of that bananas are grown in affect the VCGs present or dominance of the VCGs, such as polycultures or monocultures? So leave you with the most challenging question possibly.
Yeah, I'm not exactly sure. Maybe you can kind of like explain a little bit more. But as I've shown, it really shows that if there's just one, like the Cavendish, which are so widespread, if that is planted, then you'll find only one VCG present. And it also seems that when TR4 is introduced, it really kind of is very devastating. It takes over from other ones being present, but it really depends on what variety you are growing, and that will relate to what the VCG diversity will be, that you get. On the question of polycultures versus monocultures, maybe he could just kind of explain that question a little bit more.
Tony, would you like to explain?
Yeah, thanks, Diane. I guess I was wondering, like, if you had a backyard garden system, do you get different a different complex of VCGs in
I would,
something like a backyard garden system compared to a commercial monoculture plantation?
yeah, I would say yes, it would be different, but it depends on where this garden is. Also, I think in Indonesia or places like that where we have quite a large diversity of banana, there you will definitely see different VCGs versus other places where there's not as much VCG diversity. But in general, I would say yes, you would possibly find more VCG diversity in those type of settings.